Hi, I'm Rebecca,
I'm Ksenia.
and today we will be analyzing alcohol dehydrogenase.
Alcohol dehydrogenase was discovered in brewer's yeast in 1937 by Niglian and Wolff. It was purified and crystallized. In 1948, Wasson crystallized alcohol dehydrogenase, ADH, from horse liver, which was the first oligomeric enzyme that had an amino acid sequence and 3D structure that was determined. Later, Valley and Hodge showed zinc is a functional component in ADH, and in the early 1960s, the alcohol dehydrogenase gene was discovered in fruit flies.
So, what is alcohol dehydrogenase? Well, alcohol dehydrogenase is an enzyme that synthesizes aldehydes from alcohols. Many other mammals possess alcohol dehydrogenase, particularly those who mainly eat fruit. But our focus will be the enzyme specifically in humans and the enzyme found in the liver. There are 6 different forms of this enzyme found in humans, but this presentation will be focusing on the beta group.
The function of this enzyme is to oxidize alcohol by turning it into acetaldehyde. The acetaldehyde is then converted into acetate by aldehyde dehydrogenase that is easily utilized by our cells. Below, you can see the structural form of it.
The mechanism for this enzyme, alcohol dehydrogenase, can be seen on the left. This is the mechanism found in horse livers, but the difference between the horse and the human enzyme is that threonine 48 replaces serine 48. So alcohol dehydrogenase oxidizes the alcohol to acetaldehyde. NAD plus reduces to NADH, and the zinc ion is coordinated in active sites by cysteine 46 and 174, as well as histidine 67, which positions the alcohol group to the active site. Threonine 48 and histidine 51 deprotonate the alcohol.
The properties of this enzyme is that 1 unit of ADH converts one micromole of ethanol into acetaldehyde per minute at a pH of 8.8 at 25 degrees Celsius. It utilizes zinc and NAD plus as cofactors.
When examining the structure of alcohol dehydrogenase, the main chain has a residue of 375. The enzyme has a mass of 80 kilodaltons, and it is observed in the figure to the left that alcohol dehydrogenase is a dimeric enzyme. The main chain consists of 2 subgroups who each have a mass of 40 kilodaltons. The subunits bind 1 to 2 zinc ions.
The structure of alcohol dehydrogenase has at least 6 slightly different variants in the human body. These variants affect factors such as a more rapid conversion of alcohols to acetaldehydes. The NAD plus binding domain binds to the C terminus of the beta sheets, of residues 176 to 318. The enzyme is made up of 21 beta strands, 18 alpha helixes, and 3 terns.
So the residue can be seen below, and the residue of alcohol dehydrogenase was found using UniProt. All of the amino acids underlined in blue are hydrophobic, and those underlined in red are polar. Most of the residues in the main chain are hydrophobic and cannot bind with water.
There are 2 active sites in ADH. The zinc ion is used to hold and position the alcoholic group on ethanol with cysteine 46, 174, and histidine 67 that binds catalytic zinc ions. And then the NAD cofactor, which actually performs the reaction.
So when analyzing the catalytic activity, it converts a primary alcohol with the NAD plus into an aldehyde, a hydrogen, and NADH. These reactants and products can be seen below.
ADH catalyzes the oxidation of alcohols to their corresponding aldehydes through a mechanism of the removal of the hydrogen. This can be seen in the figure on the right. The study we focused on was the 4-amimides mimic aldehydes and inhibit liver alcohol dehydrogenase and ethanol metabolism. The purpose of the study was to determine inhibitors for alcohol dehydrogenase that would be useful in the treatment of methanol poisoning. The study accounted for the oxidation of methyl's toxic products. This was done to find uncompetitive inhibitors that could effectively control alcohol metabolism. The predictions we went into going to this experiment was anything that disrupts the hydrophobic interactions of the enzyme will make a good inhibitor. An effective inhibitor will significantly lower the rate of elimination of alcohol in the liver. The methods that was used in the study was there were 14 new furanimides were prepared and tested as inhibitors of purified liver alcohol dehydrogenase, them in horse, human, and mouse. 3 different analytical methods were used to confirm the structure of each compound: NMR, mass spectra, and gas chromatography. The synthesis of furanimides was done since furanimides are unreactive analogs of the aldehyde substrates of alcohol dehydrogenase, and they bind to alcohol and to the enzyme NADH complex and work as uncompetitive inhibitors against varied concentrations. Protein purification is a series of processes intended to isolate one protein, in the study NADH. Protein purification is the process of the characterization of the function, structure, interaction of the protein. X-ray crystallography was necessary in this study to obtain a 3D molecular structure from a crystal. The structure provides a basis for interpreting the structure-function relationship of the inhibitors. The biological evaluation of this procedure studied the effects of the inhibitors on ethanol, and the blood alcohol concentration was determined by gas chromatography.
So the results of this study can be seen in the table to the right, and it was observed that the inhibition of purified liver alcohols dehydrogenase by formamides, sulfoxides, and pyrazoles. The inhibition constants were found by measuring the change in NADH absorbance. The thiolane oxides were found to be effective inhibitors for all the enzymes. The sulfoxides were determined to be much less effective inhibitors than the thiolane oxides. The formamides were particularly effective inhibitors for the human enzymes. For our results, it was found that the inhibition constants were found for the series of N-substituted formamides, sulfoxides, and pyrazoles using initial velocity with varied concentrations of the inhibitor and substrate for each purified ADH. The formamide showed uncompetitive inhibition against varied concentrations of ethanol and competitive inhibition against varied concentrations of cyclohexanone. The alkyl formamides were better inhibitors of the mouse and horse enzymes because of water's effects on the van der Waals interactions. It was also observed that N1-methylhexylformamide decreases the rate of elimination of ethanol. The enzymes ADH between the mouse, horse, and human have a few differences of amino acid residues in the active sites and can differ by tenfold or more. RN1-methylhexylformamide, a heptyl analog, and N15-dimethylhexylformamide are particularly potent inhibitors of the human and mouse enzymes. The methylated cyclohexylformamides are better inhibitors because it has more room in the active site. The coenzyme concentrations were fixed at saturating levels, while the concentrations of substrates and inhibitors were varied. Inhibition constants were determined. It was found that the sulfoxides bind to the ENADH complex and are uncompetitive inhibitors against the varied concentrations of alcohol, whereas the pyrazoles bind to the ENAD+ complex and are competitive inhibitors. The structures of the horse enzymes complex with NADH and RN1-methylhexylformamide was determined. The geometry of binding suggests that the formamide mimics the Michaelis complex with NADH and the aldehyde. As you can see in our table, by analyzing the most effective inhibitors, it was found that the formamides were very effective, especially when compared to the oxide.
The rate of metabolism of ethanol as it pertains to the effects of the five selected inhibitors was studied with mice. The results show that the short-chain N-isopropylformamide is comparatively potent to the N-isopentylformamide. The slow rate of elimination of alcohol seen in N-isopropylformamide is a greater inhibitor when compared to the most other inhibitors. The puckering of the ring of NADH has not been seen in dehydrogenase structures with a substrate or substrate analog. The ring is puckered in the complex with NAD+ and pyrazole, which forms a partial covalent bond between carbon 4 of the ring and a nitrogen of the pyrazole and mimics the transition state of the reaction. Molecular dynamics on the native ADH and NADH complex show fluctuations of the protein cause the ring of the NADH to bend. The puckering of the ring decreases the transition state energy for hydrogen transfer at carbon 4. The inhibition consists determined in this study are thermodynamic dissociation constants that show the energies of multiple binding interactions.
Ultimately, the formamides weakly bind to the human enzymes, which results in water interfering in the hydrophobic interactions within the enzyme. Enzymes are stereoselective for the binding of inhibitors, and this can be seen at residue 141, where the mouse and human enzyme showed increased affinity when the length of the inhibitor increased because of the substitutions that occur there. The methyl alkyl sulfoxides, when measured by X-ray crystallography, are determined to be less effective inhibitors. The high affinity achieved is the result of the formation of a partial covalent bond, mimicking the transition state in the bound NAD+. Liver alcohol dehydrogenase is the predominant rate-determining factor in the metabolization of ethanol. The ADH, being the major enzyme, is a good target for inhibitory drugs. The effectiveness of inhibition depends on the metabolism of the inhibitors. The results on the rate of elimination can be used to devise treatments for methanol poisoning. The formamides are almost as potent as the competitive inhibitor 4-methylpyrazole, which is the main treatment used to treat methanol poisoning. But they are uncompetitive inhibitors against varied concentrations of ethanol. Because the formamides are uncompetitive inhibitors, this makes them effective treatments for methanol poisoning, even at high concentrations.
And here's our works cited page. Thank you.